So far, we've talked about gene expression in terms of protein synthesis. We've looked at the processes of transcription and translation and how they result in proteins at the end of it. And we've looked at understanding that that's basically what DNA does and how it controls everything about living things. What we want to look at this week is sort of how can we control these processes? We know these processes exist, but how could we control them and enhance them or calm them down, stop them completely, depending on what type of cell we're looking at, depending at the time in an organism's life. There's lots of times when we want to regulate these processes. So in this video, we're going to go through looking at gene expression, but primarily focusing on different types of gene regulation. Having a look at the objectives here, we're about halfway through this sort of subtopic, and we've got about half to go. A lot of these are low-level cognitive verbs: recognize, identify, recall. It's going to seem like there's a lot to learn, but the main thing here is just getting an idea and being able to, as it says there, recognize, identify that gene expression can be regulated in a variety of different ways. And that's the main thing we want you to take out of this in terms of understanding how this process of gene regulation occurs. Just quickly, if we have a talk about this, because I feel like I may have glanced over it recently, coding and non-coding DNA. So in terms of our transcription, we already talked about coding and non-coding DNA in terms of coding DNA being the exons, and they're the ones that eventually get translated to form our proteins, and non-coding DNA in our transcription was introns. They weren't really doing anything, it seemed. They used to be called junk DNA, but we know better than that now. They have an incredibly important role in terms of gene regulation, which is what we're talking about this week. So coding DNA only makes up approximately 2% of your genome, whereas the rest of it regulates how that 2% is expressed, when it's expressed, how often, in what magnitude. As it says there, turned on, turned off, how much, how it gets packed into the chromosomes. So those non-coding DNAs have an incredibly important role in gene regulation, those sequences. So we have our non-coding DNA, which is effectively coding for RNA or functional RNAs. That's our tRNAs or rRNAs. So these aren't being translated. They do need to be transcribed to create these RNAs, which then allow for the expression of our mRNA. So they allow for translation to occur. So that's one type of non-coding DNA. So non-coding doesn't mean it doesn't have a job. It just means its job isn't to make proteins. We have centromeres and telomeres as well, which are segments of non-coding DNA. Centromeres, we should remember from meiosis, they are the little belt around these sister chromatids which hold them together. That's their role. Pretty straightforward. Telomeres are extensions on the end of DNA, as you can see at the ends of our chromosomes. You can see in this picture. They act as caps on the end of the chromosomes to extend their life and to help protect them from damage. So they're protective. And the shortening of telomeres has been directly linked to aging. They found that as people age, they tend to have shorter and shorter telomeres. And some people seem to think that if we could solve the shortening of telomeres, we might be able to stop aging altogether. So that's our non-coding and coding DNA. Both of them are important when we're looking at gene regulation, because the coding DNA is being expressed, the non-coding DNA is often the one that's regulating the process. So just to summarize what we've talked so far about, gene expression is that process of taking our coding DNA and turning it into a protein product. That is the purpose of gene expression. It's to take DNA and turn it into a protein, which is then what we can use for all sorts of living things. all sorts of different biochemical processes, etc. So we can take our DNA wound up as a chromosome, unwind it, go through transcription. We can process that mRNA to make mature mRNA. It then goes off, is translated into a polypeptide chain, which is processed to form our active protein. And the purpose of this, well, the purpose of gene expression is to make those proteins. When we think about our DNA, though, we should be able to recall that every single one of our cells, as a human, every single one of our cells has the same DNA. There's no different genetic code for, say, a skin cell versus a heart muscle cell versus a sperm cell. They have all the same genes. Well, sperm cells obviously having half, but they have all the same genes. They have all the same DNA, but we don't need all of them. A great example here is that if you've got a liver cell, it doesn't need to know how to make neurotransmitter proteins because those only have a function inside of the brain. Similarly, a nerve cell does not need to know how to make alcohol dehydrogenase because it's not its purpose to break down alcohol. So certain cells will only need to perform certain functions. And what that means is that they'll be able to turn off any genes for proteins that don't serve their function. And those non-required genes will become inactive. This saves our cells energy and resources, which is incredibly important because it makes us much more efficient. So if a gene is then being expressed in that cell, we say that it's switched on. But a lot of genes will be switched off. Not only can we control whether a gene is switched on or switched off, which is a big part of gene regulation, controlling the expression of it, we can also control the speed at which it's transcribed and translated. So you need something happening fast, we can make that happen. Gene regulation, which is what we're going to be talking about for the rest of this, is the processes within the cells that enable these genes to be expressed just in particular cells and at certain specific times. We do have in our bodies, or in all eukaryotes, that any gene is by default switched off, which means that any cell that does need it needs to be able to switch it on. The only exception to this are housekeeping genes. Housekeeping genes are so named because they are the ones that are absolutely crucial to keeping our house basically functioning, our bodies functioning. So they control traits such as proteins and enzymes that are needed for cellular respiration and other critical biochemical processes. They code for essential macromolecules. They stop us from dying. There's a huge list of things that they do. But effectively, they are the ones that we need above anything else to stay alive. So in terms of gene regulation, we can regulate gene expression at basically any point in that process. So right from the very beginning, where our DNA is still wound up around the in chromatin, or right at the very end when we've got our protein already, we can still regulate it. To give you a brief sort of overview there, they can be regulated by chromatin accessibility. There's a first step, which is basically stopping the DNA from unwinding around the unwinding from the histones, so that if we can't unwind that DNA, we can't read it to transcribe it. Okay, so chromatin accessibility is the first opportunity to basically stop gene expression or regulate gene expression. Where the vast majority of gene regulation happens in eukaryotic organisms is with transcription factors. Transcription factors specifically control the transcription process, and we'll look at those in a bit more detail, but they are the key part of this. We could look at RNA processing as well. Our best example of RNA processing is our splicing of pre-mRNA to make our mature mRNA, okay, but also putting the guanine cap and the poly-A tail onto our mature mRNA. That's all part of RNA processing. We've got micro RNAs or miRNA, and they primarily can act on the mRNA between transcription and translation or at the point of translation. So they're effectively acting to regulate the translation of a transcribed bit of mRNA. Okay, so they're acting in that translation phase. Lastly, we have post-translation regulation, which involves affecting the protein somehow after they've already been produced. A lot of proteins, maybe not a lot, but there's a fair few that actually won't do what they're meant to do until they've gone through some sort of post-translational process. And so by stopping that, we can regulate our gene expression. Okay, these are all effectively internal factors. There are other factors that affect gene regulation as well. And the study of that is epigenetics, epi generally meaning above. Epigenetics is the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. So regulation of gene expression is not making any change whatsoever to the genetic code. We're not altering the DNA of that organism in any way whatsoever. We're just controlling what can and can't be expressed. We're not making any changes at all. Okay, changes might be like going into a document and completely editing it, taking out paragraphs, putting in your own. Whereas epigenetics is more like just only reading selected paragraphs. This epigenetics can be influenced by extracellular cues, such as developmental cues, the release of hormones in the body, such as release of, say, testosterone for a genetically male person at the beginning of puberty can cause epigenetic changes, such as growth of body hair, growth of pubic hair, dropping of the voice, etc. Those sorts of traits. Environmental factors can affect it as well. Best example being getting a tan, okay, getting a suntan. But environment also can includes anything basically outside of the body. So if you eat too much, it can have an effect on your genetic code, or if you eat too much of the bad foods, if you smoke, if you take drugs. These are all classified as environmental factors. And then other signals as well. And we can also inherit certain epigenetic things that are going on as well. So regulation of phenotype, we've talked, this is basically just leading us into talking about our different types of gene regulation. So when we're talking about regulation of phenotype, that's our epigenetics, because we're not changing the genotype, we're just controlling what the end result of expressing, if any expression happens of those genes. And as I said, most of it, the vast majority of it is happening in transcription, because that's the easiest and most efficient place for it to happen. But it can happen at any of those stages, as we said. Looking at the first factor there, chromatin accessibility. So we know that DNA is tightly coiled around histone proteins to make chromatin, and that as it's wound up there, it's not accessible to RNA polymerase. So if it's not accessible to RNA polymerase, we can't have transcription. It's as simple as that. So it needs to be uncoiled in order for transcription to occur. An acetyl group, which for those of you in chemistry is a 2-carbon chain, can be added to the chromatin, which allows the histones to relax so that transcription can occur. That is basically switching on the gene. We can have the opposite of that, which is methylation, which is where a methyl group, a single carbon and its associated hydrogens, are added to the amino acids of the histones, which causes them to be tightly wound and blocks transcription. You can see it summarized here in this diagram. So we've got histones that are positively charged, the DNA is negatively charged. If we want to open that up so transcription can happen, we need to add some acetyl groups to the histones, and that will result in them being negatively charged and will open up the chromatin. Or we add a methyl a methyl group to the histones, which makes it more positively charged, which causes it to hold that the DNA more tightly around that around the histone proteins. So the way I always remember it as acetyl on, methyl off, taking a little bit of inspiration from Mr. Miyagi there. Acetyl on, methyl off. Okay. If we want an example of this, we can have X inactivation happening in cats, which can result in such traits like this tortoiseshell coat coloring here, where one of the two X chromosomes in females is silenced by methylation, and we end up with different coat colors as a result. So long story short, we can have that methylation causing gene inactivation, which means transcription doesn't occur. Okay, it can be a good thing or a bad thing. It can be used to silence a gene that doesn't need to be used, or to prevent overproduction of a particular gene if we don't want too much of whatever the gene product is. Okay, just to show you further example here. Okay, acetylation activating the gene, and deacetylation, also known as methylation, deactivates the gene. Okay, so you can see that often we will have that acetylation methylation happening onto the histone protein, but methylation can occur directly to genes as well. So that would mean that even if transcription were able to access the DNA, it wouldn't be able to read it. All right, so we can have methylation of either the histone or the DNA. Moving on to the more important one, transcription factors. These play an incredibly important role, as I said, because they effectively allow transcription to occur or not occur. They are proteins, transcription factors, or TFs, and these proteins determine which genes are active in each cell of your body. So the example before where we had the liver cells with alcohol dehydrogenase gene active and the neurotransmitter hormone gene inactive, that is controlled by transcription factors. So in eukaryotic organisms, RNA polymerase can only attach and begin transcription at a promoter. The promoter comes immediately before the gene that we actually want to transcribe. So the promoters will have binding sites for RNA polymerase. A key example of a promoter in eukaryotic organisms is a Tata box, Tata, and you can see that it's in that sort of promoter area before we have our actual gene. Mutations in this area can be pretty bad because it is such an important part in allowing for that gene expression. So transcription factors can act as activators where they allow the DNA to unwind more easily, or they attach right at the binding site for the activator. The activator binds to the DNA where RNA polymerase is going to attach to the promoter. So it attaches there and basically makes it easier for RNA polymerase to come in. All right, so it's allowing that process to activate a lot more easily. We can also have repressors, which will bind to the promoter and completely block RNA polymerase from being able to bind there. So that stops transcription. So activators bind to the promoter to make it easier for RNA polymerase to attach, and repressors bind to the promoter to stop RNA polymerase from attaching. We can also have other types of transcription factors, such as enhancers and silencers, but these are the main two that you need to worry about. So if we have an activator, then that means that gene is going to be transcribed, which can then be translated and we make our gene product, which is our protein. But if we have a repressor, then no transcription is going to be able to occur. Okay. I talked briefly about external factors or environmental factors influencing gene regulation. One big bit of evidence that we have for this is twin studies, where, and it's not ethical anymore and they don't really do it anymore, but where you could take two identical twins which have exactly the same DNA at the beginning of their life and separate them and see how they change over their lifetime, because the environment has a significant effect on that. The nature versus nurture side of things. You could have two identical twins, and if one of them overeats bad foods, then they could end up with diabetes, okay, with those traits being expressed in them, whereas the other may not. You could end up with one with a tan and one with not. And a lot of this is due to different amounts of methylation of the DNA, which can create differences in their original DNA, which results in epigenetic changes. There are a lot of evidence of this occurring, and there's even evidence of siblings that are quite similar, differing a lot over time. Other examples as well, we've got the agouti gene in mice, which can look at how diet affects gene expression. So the agouti gene leads to them having a yellow coat, but it also makes them obese, gives them diabetes and cancer. But if they have a diet that is rich in methyl group vitamins, then that can actually stop that gene. So you can see that the pregnant mother here is fed with methyl-rich foods, and their offspring are going to be predominantly brown and in good health. Whereas if they're fed with regular mouse food, then we end up with a lot of big, fat, diabetic mice. The Himalayan rabbit has genes that can express darker pigment. Ideally, it's white because it's blending in with snow. But white isn't very good for absorbing heat. So it has dark pigments that can be switched on by extreme cold temperatures that allows them to get darker pigment in their extremities to provide them with extra warmth. So those parts of their body don't get quite so cold and they don't end up losing their fingers and their toes and their nose and their ears every winter. Then we've got prokaryotic organisms, which are very good at regulating their genes based on their environment. And we're going to talk very briefly about operons, because prokaryotic organisms don't tend to regulate their genes in the same way that we do. So when we're talking about gene transcription in prokaryotic organisms, they don't tend to have junk DNA. Instead, their transcription, their gene regulation comes primarily from their environment. So an operon, which we find in these prokaryotic organisms, is a cluster of genes required for the same purpose that are transcribed on a single mRNA strand. They're a cluster of genes that are all aiming to do the same thing. They will all have one promoter, okay, at the beginning, so they're regulated together. So that means that you have all of them on or all of them off. You can't control them individually. The two main studied examples of operons are the lac operon, which encodes for the enzyme to metabolize lactose when it's present. So by lactose being present in the environment of this bacteria, say you dump some E. coli into a nice big cup of milk, then it can switch on the genes that are able to produce the enzyme lactase, or different types of enzymes that break down lactose. I certainly wish I could do that. The trip operon is very similar in that it's an operon controlled by environmental cues, but it will produce enzymes that synthesize tryptophan, okay, as opposed to synthesizing enzymes there. So the example here of our lac operon, so when this E. coli bacteria is in an environment that contains lactose, it can switch these genes on. And you can see in the image, we've got three different genes here. Okay, one for beta-galactosidase, beta-galactosidase permease, and beta-galactosidase transacetylase. So these three enzymes are coded by these three genes that are all controlled together, which pretty much makes sense because you only need one of them if you need all of them when lactose is present. So when lactose is present for this operon to be triggered, then what happens is we have the lactose binds to the repressor. So we almost have like a permanent repressor here. And lactose binds to the repressor, which changes its shape and stops it from binding to the promoter. So it basically removes the repressor factor there. Okay, then transcription can occur and these enzymes are made, and the E. coli can go to town, chowing down on those delicious lactose molecules. When there's no lactose present, then the repressor binds to the promoter and stops the gene being transcribed. Okay. miRNAs, microRNAs, we talked about before. Okay, so microRNAs are acting at translation, and they can bind to mRNA prior to translation, after it leaves the nucleus before translation, and stop a ribosome from being able to be translated entirely. So it basically just blocks it. It binds to the mRNA, which stops that translation from occurring, and it can also lead to the destruction of the mRNA, which is usually comes in the form of it being basically chopped up. Okay, and miRNAs, microRNAs are genes themselves, will come from genes themselves, I should say, because they're functional RNAs. So they can be regulated as well. So it's sort of like a two-step process, really. That's the majority of that sort of thing in terms of gene regulation. What we want to look at now is particularly some examples of morphology and other regulation in organisms. And I'm going to go through this quite briefly, but you can look at it in more detail. The two specifically mentioned by the syllabus are HOX genes or homeobox genes and SRY genes. So these genes are regulating growth and development, and as I said earlier, they can be switched on and off at certain times. So if we look in particular at one of our best friends when genetics is concerned, Drosophila melanogaster, it's often used in genetic studies because it has quite a simple and easily analyzable and easily investigated genetic code. Its genome is easy to work with. So it has 13,000 genes, but only 8 of those genes code for its basic body structure. So we call those homeobox or toolkit genes, HOX genes for short, and they're genes that code for the proteins that regulate body formation and patterning in the developing embryo. So you can see on this diagram here the 8 genes, the 8 HOX genes for our flies. We have these in all organisms though. Humans have HOX genes also that code for how we develop. And if there's any developmental issues in terms of people being born with the wrong limbs or that sort of thing, or without limbs, then that can often be attributed to problems that affect the homeobox genes. Our other one, as I said, is SRY genes. SRY stands for sex determining region Y protein. And that protein is involved in male sexual development, which is determined by the chromosome an individual has. So people who are genetically female will have 2 X chromosomes. So they have an XX karyotype, while people who are genetically male will have an X and a Y chromosome. On that Y chromosome, we have the SRY gene, okay? It's only found on the Y chromosome. What happens is you've probably all heard that idea that at some point in the womb, everyone is a female. So this SRY gene produces a protein which acts as a transcription factor for specific regions of DNA that result in male development. So effectively, the SRY gene is the switch for male sexual development, such as the fetus developing gonads, male gonads, testes, and preventing female reproductive structures such as a uterus or fallopian tube from being developed. So these genes are the transcription factors that control male sexual development. If we have a look at chickens, for example, we have male or female eggs. They develop a genital ridge, and they have DMRT1, which potentially acts to sexually differentiate them. Whereas with males, we have the SRY. So we have an egg that forms a genital ridge and indifferent gonads. So gonads are just your sexual reproductive organs, and at this point, they could be ovaries, they could be testes. There's no determination there. The SRY gene either kicks in or it doesn't. And if it does kick in, those indifferent gonads form testes, which then produce hormones, which produce the typically male phenotype. Whereas if the SRY gene doesn't kick in, then it goes to ovaries, which produce hormones, which produces the female phenotype. And that's basically it. Now, I know that was a lot to take in, but remember, it all comes down to the objectives. So recognizing the purpose of gene expression is to synthesize a functional gene product, and this process can be regulated. Identifying factors that regulate phenotypic expression, 3 mentioned there, is transcription and translation. Okay, that it can be regulated through products of another gene or via environmental exposure. Recognizing that differential gene expression regulates cell differentiation, and then recall an example of a transcription factor that regulates morphology. The example we looked at is the Hox factor, and then a transcription factor that regulates cell differentiation is our SRY, or sex determining region Y. Okay, that is it. I know it was a long one, but hopefully you've taken it all in. If not, have a read through. Also, maybe check out Khan Academy and Amoeba Sisters. They've got some great videos on this. Thank you.